Hi class, this is your instructor, Skylar Huff. We're now in Chapter 11. This is Sexual Reproduction and Meiosis. Most animals and plants reproduce sexually. Gametes of opposite sex unite to form a cell that, dividing repeatedly by mitosis, eventually gives rise to an adult body with some 100 trillion cells. The gametes that form the initial cell are the products of a special form of cell division called meiosis. If you look in your text, you will see this photo, and as you see this photo, this is what it looks like. So meiosis is far more intricate than mitosis, and the details behind it are not as well understood. The basic process, however, is quite clear. Also clear are the profound consequences of sexual reproduction. It plays a key role in generating the tremendous genetic diversity that is the raw material for evolution. So we'll go through these 4 sections of the chapter, and let us begin. The essence of sexual reproduction is genetic contribution of 2 cells. This genetic contribution is the mode of reproduction imposes difficulties for sexually reproducing organisms that biologists recognized early on. We are only recently making progress on the underlying mechanism for the elaborate behavior of chromosomes during meiosis. To begin, we briefly consider the history of meiosis and its relationship to sexual reproduction. Only a few years after Walter Fleming's discovery of chromosomes in 1879, Belgian cytologist Eduard van Beneden was surprised to find different numbers of chromosomes in different types of cells in the roundworm from the genus Ascaris. Specifically, he observed that the gametes, the eggs and sperm, each contained 2 chromosomes, but all of the non-reproductive cells, or somatic cells, of embryos and mature individuals contained 4. From his observations, Van Beneden proposed in 1883 that an egg and sperm, the gametes, each containing half the complement of chromosomes found in other cells, fuse to produce a single cell called a zygote. The zygote, like all cells ultimately derived from it, contains 2 copies of each chromosome. The fusion of gametes to form a new cell is called fertilization, or syngamy. It was clear even to early investigators that gamete formation must involve some mechanism that reduces the number of chromosomes to half the number found in other cells. If it did not, the chromosome number would double with each fertilization. After only a few generations, the number of chromosomes in each cell will become impossibly large. For example, in just 10 generations, the 46 chromosomes present in human cells would increase to over 47,000. The number of chromosomes does not explode in this way because of a special reduction division. It is referred to as meiosis. Meiosis occurs during gamete formation, producing cells with half the normal number of chromosomes. The subsequent fusion of 2 of these cells ensures a consistent chromosome number from one generation to the next. Meiosis and fertilization together constitute a cycle of reproduction. 2 sets of chromosomes are present in the somatic cells of adult individuals, making them diploid cells, but only 1 set is present in the gametes, which are thus haploid. Reproduction that involves this alternation of meiosis and fertilization is called sexual reproduction. Its outstanding characteristic is that offspring inherit chromosomes from 2 parents, which you all will see here in figure 11.1. You, for example, inherit 23 chromosomes from your mother, maternal homologue, and 23 from your father, paternal homologue. The life cycles of all sexual reproducing organisms follow a pattern of alternation between diploid and haploid chromosome numbers. But there is some variation in life cycles. Many types of algae, for example, spend the majority of their life cycle in a haploid state. The zygote undergoing meiosis produces haploid cells that then undergo mitosis. Some plants and some algae alternate between a multicellular haploid phase and a multicellular diploid phase. I won't teach those in this course, but those will be taught in Biology 2, or Principles of Biology 2, In most animals, the diploid state dominates. The zygote first undergoes mitosis to produce diploid cells. Then later in the life cycle, some of these diploid cells undergo meiosis to produce haploid gametes, which you all see here in figure 11.2. So as you have it, the diploid adults are there. By way of meiosis, each of those diploid adults produce the gametes, the sperm and egg. By way of fertilization, the diploid zygote is formed. By way of mitosis, we go back to that, I guess you shall say, later in life, or nearly later in life, not quite an adult, a child. In animals, the single diploid zygote undergoes mitosis to give rise to all the cells in the adult body. The cells that will eventually undergo meiosis to produce gametes are set aside from somatic cells early in the course of development. These cells are referred to as germ-line cells. Both somatic cells and germ-line cells are diploid. The critical difference is that somatic cells undergo mitosis to form genetically identical diploid daughter cells, but germ-line cells can undergo meiosis to produce haploid gametes. So that is why you're seeing in the center left those two examples of germ-line cells, or at least where the germ-line cells lie, and even that so at that bottom right-hand side. The mechanism of meiotic cell division varies in important details in different organisms. These variations are particularly evident in the chromosomal separation mechanisms. Those found in protists and fungi are different from those in plants and animals, which we describe here. Meiosis in a diploid organism consists of two rounds of division called meiosis 1 and meiosis 2, with each round containing prophase, metaphase, anaphase, and telophase stages. Before describing the details of this process, we first examine the features of meiosis that distinguished it from mitosis. During early prophase 1 of meiosis, homologous chromosomes find each other and become closely associated, a process called pairing or synapsis. Let's show synapsis. This is more or less synapsis. Despite a long history of investigation, molecular details remain unclear. Biologists have used electron microscopy, data from genetic crosses, and biochemical analysis to shed light on synapsis. Thus far, the results of their investigations have not been integrated into a complete picture. It is clear that homologous chromosomes find their proper partners and become intimately associated during prophase 1. This process includes the formation of many species of an elaborate structure called synaptomenal complex, or synaptomenal complex, joining the paired homologues. The synaptomenal complex consists of a central element connected by filaments to two sets of lateral elements that interact with the homologues. The structure of synaptomenal complex appears similar in all systems that have been examined, although its exact function is unclear. A representative example is shown in figure 11.3b. This results in the paired homologues, each composed of two sister chromatids, all being closely associated during this phase of meiosis. The structure is called a tetrad or bivalent, and I refer to them as tetrads. This is that synaptomenal complex spoken to moments ago. While homologs are paired during prophase 1, another process unique to meiosis occurs: genetic recombination, or crossing over. This process literally allows the homologs to exchange chromosomal material. The cytological observation of this phenomenon is called crossing over, and its detection genetically is called recombination, because alleles of genes that were formerly on separate homologs can now be found on the same homolog. The sites of crossing over are called chiasmata, and the sites of contact are maintained until anaphase 1. The physical connection of the homologs due to crossing over, and the continued connection of sister chromatids lock homologs together. The association between the homologs persists throughout meiosis 1 and dictates the behavior of the chromosomes. During metaphase 1, the paired homologs move to the metaphase plate, and they then become oriented with homologs of each pair attached to opposite poles of the spindle. By contrast, in mitosis, the homologs do not associate and behave independently of one another. Then, during anaphase 1, homologs are pulled to opposite poles for each pair of chromosomes. This again is in contrast to mitosis, where sister chromatids and not homologs are pulled to opposite poles. This is why the first division is termed the reduction division. It results in daughter cells that contain one homolog from each chromosome pair. The second meiotic division does not further reduce the number of chromosomes. It will merely separate the sister chromatids for each homolog. The most obvious distinction between meiosis and mitosis is the simple observation that meiosis involves two successive divisions with no replication of genetic material between them. One way to view this is that DNA replication must be suppressed between the two meiotic divisions. Because of the behavior of the chromosomes during meiosis 1, the resulting cells contain one replicated copy of each chromosome. A division that acts like mitosis without DNA replication converts these cells into ones with a copy of each chromosome. This is the last key to understanding meiosis. The second meiotic division is like mitosis with no chromosome duplication. To understand meiosis, it is necessary to carefully follow the behavior of chromosomes during each division. The first meiotic division depends on each homologous pair behaving as a unit and not as individual chromosomes, as they do in mitosis. This is accomplished by a complex set of processes that join homologs until anaphase 1 when they are separated. Meiotic cells have an interphase period that is similar to mitosis with G1, S, and G2 phases. After interphase, germline cells enter meiosis 1. In prophase 1, the DNA coils tighter, and individual chromosomes become visible under the light microscope as a matrix of fine threads. Because the DNA has already replicated before the onset of meiosis, each of these threads actually consists of two sister chromatids joined at their centromeres. In prophase 1, homologous chromosomes become closely associated in synapses and exchange segments by crossing over, which leaves all four chromatids associated. During interphase in germline cells, the ends of the chromatids seem to be attached to the nuclear envelope at specific sites. The sites the homologs attach to are adjacent, so that during prophase 1, the members of each homologous pair of chromosomes are brought close together. Homologous pairs then align side by side in the process of synapsis. Synapsed homologs are joined along their entire length. The sister chromatids of each homolog are also joined together by cohesin proteins. This association of sister chromatids is called sister chromatid cohesion. Sister chromatid cohesion also occurs during mitosis, but in meiosis, the cohesin complex contains a meiosis-specific cohesin that is important for later events. Along with the synaptominal complex that forms during prophase 1, another kind of structure appears at the same time that recombination occurs. These are called recombination nodules. and they are thought to contain an enzymatic machinery necessary to break and rejoin chromatids of homologous chromosomes. Crossing over involves a series of events in which DNA segments are broken and then rejoined at the same site, exchanging material between the homologs. The molecular events that break and rejoin homologs are controlled such that each chromosome arm usually has one or a few crossovers per meiosis. Human chromosomes typically have 2 or 3. After crossing over occurs, the synaptimal complex breaks down. Homologous chromosomes are less tightly associated, but remain attached by chiasmata. At this point, each chromosome is organized into a tetrad with 4 chromatids, and it does say bivalent, but I said tetrad. So 2 homologous, or at least 2 homologs, each with 2 sister chromatids. And that's where we have it, right there in the center, the tetrad, or the bivalent. The 4 chromatids are held together in 2 ways. 1, the sister chromatids of each homolog, the products of DNA replication, are held together by cohesin proteins. That's sister chromatid cohesion. And secondly, the exchange of material by crossing over between homologs locks all 4 chromatids together, as you see here in figure 11.4. While the elaborate behavior of chromosome pairing is taking place, other events must occur during prophase 1. The nuclear envelope must be dispersed, along with the interphase structure of microtubules. The microtubules reform into a spindle, just as in mitosis. By the end of prophase 1, each homologous pair is locked together as a tetrad. These tetrads capture spindle fibers and move to the center of the cell and align on the metaphase plate as paired homologs. This alignment of homologs occurs because the kinetic cores of sister chromatids for each homolog act as a unit to capture polar microtubules. Therefore, microtubules from opposite poles are attached to the homologs and not sister chromatids, as occurs in mitosis. We call this monopolar attachment, as each homolog is attached to one pole instead of having its sister chromatids attached to the opposite pole in the bipolar fashion of mitosis. The molecular basis of monopolar attachment is not understood. One model with some support is that the centromere kinetochore complex of sister chromatids is compacted during meiosis 1, allowing them to function as a single unit. This monopolar attachment of kinetochores of sister chromatids would be disastrous in mitosis, but it is critical to meiosis 1. It produces tension on the paired homologs, pulling them to the equator of the cell. In this way, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may be oriented toward a given pole. This also provides random variation. It makes us different. So this is showing a random orientation of chromosomes, such as you see in figure 11.6. In anaphase 1, the microtubules of the spindle fibers begin to shorten. As they shorten, the connections between homologs at chiasmata are broken, allowing homologs to be pulled to opposite poles. Anaphase 1 comes about by the release of sister chromatid cohesion along the chromosome arms, but not at the centromeres. This is the result of the destruction of meiosis-specific cohesin in a process analogous to anaphase in mitosis. The difference is that the destruction is inhibited at the centromeres by a mechanism that will be discussed in the next chapter. As a result of this release, the homologs are pulled apart, but not sister chromatids. Each homolog moves to one pole, taking both sister chromatids with it. When the spindle fibers have fully contracted, each pole has a complete haploid set of chromosomes consisting of one member of each homologous pair. This is that reduction division. Because of the random orientation of homologs, or at least of homologous chromosomes, on the metaphase plate, a pole may receive either the maternal or paternal homologue from each chromosome pair. As a result, the genes on different chromosomes assort independently. In other words, we're getting to independent assortment. That is, meiosis 1 results in independent assortment of maternal and paternal chromosomes into the gametes. This here is yet another way meiosis provides variation. By the beginning of telophase 1, the chromosomes have segregated into two clusters, one at each pole of the cell. Now the nuclear membrane reforms around each daughter nucleus. Because each chromosome within a daughter nucleus had replicated before meiosis 1 began, each now contains two sister chromatids attached by a common centromere. Note, the sister chromatids are no longer identical because of crossing over that occurred in prophase 1. As you will see, this change has important implications for genetic variability or genetic diversity. Cytokinesis, the division of the cytoplasm, and its contents may or may not occur after telophase 1. The second meiotic division, meiosis 2, occurs after one interval of variable length. Segregation can occur without crossing over. And let me pause and go back here. The preceding description of meiosis 1 relies on the observation that homologs are held together by chiasmata and by sister chromatid cohesion. This connection produces the critical behavior of chromosomes during metaphase 1 and anaphase 1, when paired homologs move together to the metaphase plate and then move to opposite poles. Although this connection of homologs is the rule, there are exceptions. In the fruit fly, the Drosophila, males for example, there is no recombination, and yet meiosis proceeds accurately, a process called achiasmate segregation. This seems to involve an alternative mechanism for joining homologs and then allowing their segregation during anaphase 1. Telomeres and heterochromic sequences have been implicated, but the details are not known. Despite these exceptions, the vast majority of species that have been examined use the formation of chiasmata, sister chromatid cohesion, to hold homologs together for segregation during anaphase 1. Typically, interphase between meiosis 1 and 2 is brief and does not include an S phase. Meiosis 2 resembles a normal mitotic division. Prophase 2, metaphase 2, anaphase 2, and telophase 2 follow in quick succession. For instance, prophase 2. At the two poles of the cell, the clusters of chromosomes enter a brief prophase 2, each nuclear envelope breaking down as new spindles form. In metaphase 2, spindle fibers from opposite poles bind to kinetochores of each sister chromatid, allowing each chromosome to migrate to the metaphase plate. And this is as a result of the tension on the chromosomes from polar microtubules pulling on the sister centromeres. In anaphase 2, the spindle fibers contract and the cohesin complex joining the centromeres of sister chromatids is finally destroyed, allowing sister chromatids to be pulled to opposite poles. This process is essentially the same as anaphase during a mitotic division. Telophase 2. Finally, the nuclear envelope reforms around the four sets of daughter cells. Cytokinesis then follows. The final result of this division is 4 cells containing a complete haploid set of chromosomes. The cells that contain these haploid nuclei may develop directly into gametes as they do in animals. Alternatively, they may themselves divide mitotically as they do in plants, fungi, and many protists, eventually producing greater numbers of gametes, or, as in some plants and insects, adult individuals with varying numbers of chromosome sets. One interesting variation on meiosis is observed in mammalian females, where the final products are a single egg and 3 much smaller polar bodies. So there are not 4 eggs produced per meiotic division. Errors in meiosis. So it's critical that meiosis is accurate because any failure will produce gametes with too many or too few chromosomes. The movement of chromosomes to the poles is also called disjunction. So failure of this is called non-disjunction, which produces one gamete that lacks a chromosome and one gamete that has two copies. Gametes with an improper number of chromosomes are called aneuploid gametes, and the condition is referred to as aneuploidy. In humans, this condition is the most common cause of spontaneous abortion. Implications of aneuploid gametes are explored in further detail in Chapter 13. The key to meiosis is understanding differences between meiosis and mitosis. The basic machinery in both processes is the same, but the behavior of chromosomes is distinctly different during the first meiotic division. So if you look here, this figure shows that difference. And I'll call this figure 11.5. Meiosis is characterized by four distinct features: homologous pairing and crossing over joins maternal and paternal homologs during meiosis 1. Sister chromatids remain connected at the centromere and segregate together during anaphase 1. 3. Kinetochores of sister chromatids are attached to the same pole in meiosis 1 and to opposite poles in mitosis. And fourthly, DNA replication is suppressed between the two meiotic divisions. Although the underlying molecular mechanisms are unclear, we will consider what we know of each of these features in the rest of this section. The pairing of homologous chromosomes during prophase 1 of meiosis is the first deviation from mitosis and sets the stage for all of the subsequent differences. So figure 11.8, as you're seeing now, shows this. How homologs find each other and become aligned is one of the great mysteries of meiosis. Some cytological evidence implicates telomeres and other specific sites as being necessary for pairing, but this finding does little to clarify the essential process. The process of sister chromatid cohesion is similar to mitosis, but involves cohesin proteins that contain meiosis-specific subunits. In yeast, the protein REC8 or REC8 replaces the mitotic SCC1 protein as part of the cohesin complex. So the replacement of this critical cohesin component with a meiosis-specific version seems to be a common feature in systems analyzed to date. So the molecular details of recombination process that produces crossing over are complex, but many of the proteins involved have been identified. This process is initiated with the introduction of a double-strand break in one homolog. This explains the similarity in the machinery necessary for meiotic recombination and the machinery involved in the repair of a double-strand breaks in DNA. Recombination probably first of all as a repair mechanism and was later co-opted for using disjoining chromosomes. Mutations that destroy the function of recombination proteins in many organisms also result in higher levels of meiotic non-disjunction, which indicates the recombination is involved in the normal disjunction of meiotic chromosomes. Meiosis 1 is characterized by the segregation of homologs, not sister chromatids, during anaphase. For this separation to occur, the centromeres of sister chromatids must co-segregate or move to the same pole during anaphase 1. This means that meiosis-specific cohesin proteins must first be removed from the chromosome arms, then later from the sister centromeres. Homologs are joined by chiasmata, and sister chromatid cohesion around the site of the exchange then holds homologs together. Destruction of the REC8 protein on the chromosome arms appears to be what allows the homologs to be pulled apart at anaphase 1. The co-segregation of sister centromeres requires that the kinetochores of sister chromatids are attached to the same pole during meiosis 1. This attachment is in contrast to both mitosis, and meiosis II, in which sister kinetochores must become attached to opposite poles. The underlying basis of this monopolar attachment of sister kinetochores is unclear, but it seems to be based on structural differences between centromere kinetochore complexes in meiosis I and in mitosis. Mitotic kinetochores visualized with the electron microscope appear to be recessed, making bipolar attachment more likely. Meiosis I kinetochores protrude more, making monopolar attachment easier. It is clear that both the maintenance of sister chromatid cohesion at the centromere and monopolar attachment is required for the segregation of homologs that distinguishes meiosis I from mitosis. After a mitotic division, the new round of DNA replication must occur before the next division. For meiosis to succeed in having the number of chromosomes, this replication must be suppressed between the two divisions. The detailed mechanism of suppression of replication between mitotic divisions is unknown. One clue is the observation that the level of one of the cyclins, cyclin B, is reduced between mitotic divisions, but not lost completely, as it is between mitotic divisions. During mitosis, the destruction of mitotic cyclin is necessary for a cell to enter another division cycle. The result of this maintenance of cyclin B between mitotic divisions in germline cells is the failure to form initiation complexes necessary for DNA replication to proceed. This failure to form initiation complexes appears to be critical to suppressing DNA replication. And finally, the daughter cells produced by mitosis are identical to the parental cell, at least in terms of their chromosomal constitution. This exact copying is critical to producing new cells for growth, for development, and for wound healing. Meiosis, because of the random orientation of different chromosomes at the first mitotic division and because of crossing over, rarely produces cells that are identical. The gametes from meiosis all carry an entire haploid set of chromosomes, but these chromosomes are a mixture of maternal and parental homologs. Furthermore, the homologs themselves have exchanged material by crossing over. The resulting variation is essential for evolution and is the reason that sexual reproducing populations have much greater variation than asexually reproducing ones. Meiosis is not only critical for the process of sexual reproduction, but also the foundation for understanding the basis of heredity. The different cells produced by meiosis form the basis for understanding the behavior of observable traits in genetic crosses. In the next two chapters, or at least chapters 12 and 13, we will follow the behavior of traits in genetic crosses and see how this correlates with behavior of chromosomes in meiosis. This has been your instruction.